So welcome everybody to the Atto Fridays. This is the first one of 2024. And I think we are excited since the Nobel Prize announcement last year. And we are proud and very honored today to announce our first speaker of the year, who is Professor Linda Young. She is a researcher in X-ray science at University of Chicago and Argonne National Laboratory, where she is a distinguished fellow and leads the AIMO physics group. Her group uses primarily accelerator-based light sources, X-ray free electron lasers, and synchrotrons to understand X-ray interactions with matter from the linear to nonlinear regimes. The recent development of synchronized two-color Attosecond X-ray laser pulses with microjoule energies allows to initiate and probe radiation-induced processes on the natural time and length scale. So, I mean, if we talk about making movies and seeing things that, like in real time, this is one of the ways, definitely. And she obtained her bachelor's degree from MIT and PhD from UC Berkeley. And she has served as X-ray Science Division Director of the Advanced Photon Source, Chair of the Division Atomic, Molecular, and Optical Physics of the APS. She is a fellow of the APS and has been a recipient of a Helmholtz International Fellowship and an honorary doctorate from Uppsala University. So we were talking before we started that water is very controversial, and today she's going to talk about Attosecond X-ray studies of liquid water. Maybe in 3 years, when we organize new quantum battles, it could be a topic. We haven't chosen topics yet, but we always keep an eye out there for controversial topics. So we are ready to start. Thank you all for joining. We hope you enjoy the talk. And Linda, the floor is yours. Feel free to take it.
Yeah, thank you very much, Carla. It's a pleasure to be giving the very first Atto Fridays Seminar in 2024, especially right after that Nobel Prize. And as Carla mentioned, you know, I really don't come from the pure attosecond world, but kind of backed into it with the advent of attosecond pulses from free electron lasers. And what I'll tell you about today, with some background before, is how we can actually use 2 attosecond X-ray pulses to look at even the structure of liquid water, because you can actually freeze all nuclear motion with these super fast X-ray pulses. And that's, of course, produced by the free electron laser out at LCLS with a large amount of development work behind it. But before I get into that, let me just quickly, it was so exciting about the Nobel Prize, right? And so for anything for an Atto seminar, one should really look at that. And so here, of course, was a Nobel Prize in Physics given out just on December 10th of last year for experimental methods to generate attosecond pulses of light for a study of electron dynamics in matter. And I watched these very nice Nobel lectures in physics that were presented on December 8th. And I was really surprised to see about the last slide that Pierre Agostini put up was about the X-ray free electron laser and how it might be actually the future of attosecond science. And he goes, oh, just a quick look toward the future. So far, attosecond pulses are created by visible or infrared lasers, but maybe the future of attoseconds is not in our lab, but with this X-ray free electron laser. And indeed, this is the X-ray free electron laser that I used, well, that our team used for these studies on liquid water. Okay, so let me fast forward like a month later. There was this nice colloquium, physics, the PQE colloquium, and Paul Corkin was there. And he was telling us, oh, well, you know, what are we going to do with these attosecond X-rays? And these are are quotes from him. It says, oh, well, building on a 100-year tradition of X-ray science, we integrate X-rays and laser physics, thereby extending the spectral reach of laser spectroscopy by 2 orders of magnitude. And the integration of ultrafast with subcellular biology and integrated electronics may be the most important implication of all. So I thought, oh, I really feel like I'm in the right field here. Pierre Agostini says, oh, well, we can use these, these X-ray free electron laser pulses. Paul says, oh, we could kind of get into this subcellular biology and integrated electronics. And of course, subcellular biology suggests that we should look at water, right? We are all 2/3 water. The Earth is 2/3 water. And so one should really try to understand how X-rays interact with water. And so this is kind of what we did. There are, of course, some very practical applications for this. Because the interaction of X-rays or ionizing radiation with water, of course, is important for human health, both in a deleterious way. When you go into space travel, you're subjected to lots of cosmic rays and whatnot, and of course this causes a lot of free radical production and damage in the body, but also can be used in positive ways in cancer therapeutics. But beyond health, it's also super important to understand the radiolysis of liquid water, also because it causes corrosion in nuclear power plants and repositories and whatnot. So it's really one of these very basic things you'd like to understand. What's going on when you have a radiation radiation-induced ionization and excitation in liquid water. And of course, this is not a new topic, right? And it's been studied for quite some time. There's this very nice review article by Sophie Lecaire in 2011 that talks about, water radiolysis in sort of a review. And it's basically classified in 3 stages. A physical stage, very fast stage with ionization excitation. A physical chemical stage where you have this cascade of electrons and ions and radicals that really form this basis of solution in interfacial chemistry and aqueous environments. And if you look, a little bit further down in the chemical stage, maybe after a microsecond or so, you find all these products, right? And this is what people have done for a long time. They see all these products. And the 2 major ones are the hydrated electron. It's the primary reducing radical formed in radiolysis of water. And then the hydroxyl radical, which is the primary oxidizing species formed. And you can see they outweigh the other components, which are also very important. But it turns out that the attention, and I'm sure you folks have heard about this before, has really been focused on this solvated electron. And this dates back a long time. If you take a look at this paper in JACS in 1962, it's the absorption spectrum of the hydrated electron in water and in aqueous solutions. And interestingly, it was also a contribution from Argonne National Lab. And the reason that the attention has been focused on this the solvated electron is it has this convenient strong absorption feature at around 720 nanometers. It's right in the heart of where all the ultrafast lasers have been working. But this is back in 1962 that they found it, but I just want to say that it continues to be a topic of a study, and there are many issues associated with it. There's a chemical reviews issue that's totally devoted to solvated electron, and then more recently the hydrated electron with an annual reviews of PChem. And it's been studied both in real liquids, in bulk water, but also in clusters. And so the interesting thing is with X-rays, we could not only look at the hydrated electron as you would with visible radiation, but we can also address the valence hole spectroscopically. And I will show you how we can do that. But before then, there are many unresolved issues in ultrafast radiation chemistry. There's in this primary ionization step, one can ask, well, what is the extent of the delocalization in the hole when you've ionized water? What's the time scale of this whole localization after you've, freed the electron, put it into the conduction band, and then you have this quasi-free electron. How is the solvated electron formed? What's the solvent structure around the hydrated electron, and so on and so forth. And the standard way people have been studying this for a long time has been electron pulse radiolysis experiments, and these are typically on the picosecond time scale. So they really don't have the time resolution to address these issues. So let's see. In-step X-valves, right? And they really open up this femto and attosecond regime to look at radiolysis in complex systems. And so we started our studies using sort of a proxy for ionization, using strong field ionization, something very familiar to this crowd, and then use an X-ray probe. And that's shown in these these publications here. And I'll tell you a little bit about them. And then more recently, we've been using X-ray pump, X-ray probe studies with attosecond timescales to actually freeze all nuclear motion and learn some really interesting things. Okay, I won't talk about this today, but of course, X-rays, well, at least X-rays produced by free electron lasers are super flexible. And so you can produce X-rays all across the spectrum. And so you can imagine actually looking at these ionization processes in very complex systems. And that's part of an energy frontier research center that I'm involved in. Okay, so let's take a quick look. So here's one way that you can start to look at ionization processes in liquid water. And if you do strong field ionization, you basically remove the outermost an electron from the HOMO, the 1b1, and then that produces a hole. And this hole can be repopulated with an X-ray that's tuned right to that resonance. And so what you can see in this graph on the right is that hole is very far away from the standard liquid water absorption spectrum. So here, liquid water, if you want to look at it with X-rays, you will see an absorption spectrum that looks like this gray stuff over here on the right. And then, and if you now produce this hole, it's over here, 10 eV away or so. And so it's in this very clean so-called water window. And so we have this nice soft X-ray probe that can look at both the H2O plus as it's initially formed and also the hydroxyl radical that will be formed after, for example, a proton transfer event happens. So the way we did this experiment, it was also at the LCLS. You would have your strong field ionization pulse coming from a standard TISAF laser from which you can control the polarization. Then you'd have your X-ray pulses to probe what your sample afterwards. And then you would detect both by transient absorption and also by X-ray emission. Now, it was quite important to have this liquid microjet because, in fact, the transmission in soft X-rays through a jet, it must be quite thin. So this was on the order of a few microns or so. And so when we did this, we could take a look at the absorption before and after ionization, and that's what's shown here. And lo and behold, just as we had predicted theoretically, yes, indeed, we do see this valence hole popping up before you have this big water absorption spectrum. And so you can sit around this area and follow in time what's going on, and that's what's shown over on the right here. And you can fit what's happening as a function of time. And what we found were sequential kinetics for all three processes. So it was on the order of 46 femtoseconds for proton transfer because, oops, if you make this this hole in this this H2O plus, it's very near a bunch of other waters, and so you can have a rapid proton transfer. Then there's a vibrational relaxation and eventually a recombination. And so what you can see here, and I want to point out also this little edge shift that we observed that we'll come back to. So what you can see is that if you do the theory, and Robin Santro's group did the theory for us associated with this, is that when you watch these X-ray spectral changes after this impulsive ionization, what you can see is that as you evolve in time, there is a small spectral shift that's this red line that's on the order of a few tenths of an eV that correlates with the distance between the oxygen atom nearest the of the ionization and that on which proton transfer occurs. So that's kind of this purple line here. And so you can see that they track each other such that these X-ray spectral changes really reflect the environment around this valence hole. Okay. So then we wanted to go back to look, what's going on with that edge shift. If you remember here, our jet was too thick. And so we couldn't really see the true water absorption spectrum. So we came back a couple of years later at a completely new end station. and we had a thinner jet even. And we did the first coordinated undulator monochronometer scan, and we got this full water X-ray absorption spectrum between 500 and 550 eV. And I was so delighted when we could actually make a water absorption spectrum look basically exactly like one that was obtained with synchrotron radiation by Philip Vernet and his colleagues previously. And most people thought, oh, why are you so excited about this? And well, actually made something that looks like something that you would trust from a synchrotron at an FEL. So we were very pleased with that. Okay. And so then after that, we said, okay, we can actually make a pure water absorption spectrum, but now we can also do the ionization there, and we can actually look for all these products, right? So this is based on top here. This is the proton transfer reaction that we're looking at. This is the H2O plus a nearest neighbor water, and this quasi-free electron eventually goes to the hydroxyl radical, transfers a proton, the H3O plus, and this hydrated electron. And so if you look after 2 picoseconds, which is basically the time for solvation, you can see that the changes that you see in the spectrum are captured by these individual contributions. There's this big OH peak in the water window, and then there's this contribution from the solvated electron, and then out here, way in the higher energy region, is a signature of this H3O plus. And so this was reported in this recent article in JACS that also did a lot of theory on, how is it indicates that there's actually a cavity around the solvated electron, and how is that cavity actually formed? And there's some very nice, sophisticated calculations using ring polymer molecular dynamics that show how this happens, and it's temperature dependent. So that's very nice, and you can read about it if you're interested. Okay, but now let's go to the Attosecond regime, right? Because I guess you guys are particularly interested in attoseconds, right? And so with the attoseconds, we can do two attosecond X-ray pulses. We can actually do not just the outer valence ionization, but we can actually also do a full valence ionization with an attosecond pump. And then we can also probe in that same region that we were looking before, so at around 500 eV near the oxygen K edge, and look for all the changes in the electronic structure on this super fast time scale before any nuclear motion. So we did this first attosecond X-ray pump, X-ray probe experiment in liquid water. This was back in April of 22. And so we had this valence ionization pump. which could eject electrons from any of these valence orbitals. And then we could have this scanning core to valence pump where we could actually look at what we had created. And so we term this thing AXATAS for all X-ray attosecond transient absorption spectroscopy. And basically what you have are these 2-color attosecond X-ray pulses, which in principle can be delayed by up to about maybe 100 femtoseconds or so. So first you have your valence ionization pump, and then you have your core to valence probe. You can control the relative intensities of them with a gas attenuator. It goes into your sample, and it's a super simple configuration for just attosecond transient absorption. So the pump and the probe go through the sample, some filters and a slit, and then are basically dispersed on a 2D camera. And so this is a particularly powerful configuration that's been used a lot in attosecond spectroscopy, because the time resolution can be determined basically by the pump and probe pulse durations, and the energy resolution, and it's decoupled from the energy resolution, which is determined by this transmission spectrometer. And so for our experiment, we had these pulses coming from the LCLS that were on the order of 600 attoseconds. And it was important that it was generated in a so-called omega-2 omega mode. This was pioneered by Aggo Marinelli, Sichi Lee, and James Cryan, because this omega-2 omega mode allowed to reuse the microbunching in the first undulator series in the second undulator and get very small time delays. So we had this pump pulse at omega between, say, 250 eV or so with very lowish pulse energies. And then we had a probe pulse, scanning around 500 eV. And so they would scan together in unison. And then we had a small target, and we had this 2D detection with this grading spectrometer. And so all those things were important. But since this was the very, very first attosecond transient absorption, all attosecond X-ray pump probe, we really need to understand, what is the nature of the signal that we're actually looking at? Well, what happens when what happens when the attosecond X-ray pulse interacts with the water to create the full valence ionization, and what do you actually see on the detector? And so there are a couple of things that are important. One is this effect of electron impact ionization and coherence associated with that. And it turns out that electron impact ionization plays a very large role in what you actually see. Because so let's say you come in with a 250 eV pulse and you ionize your valence electron, you end up with an electron that's around 200 eV or so. And the electron that's around 200 eV is going at about 3% the speed of light. So it'll travel on the order of 10 nanometers or so before it within 1 femtosecond. And so then the mean free path of an electron of that energy is only on the order of a nanometer or so. And so you're going to have all of these electron impact ionization collisions that are also impacting your sample during the course of the pump pulse and also the probe pulse. So here is a pump pulse and a probe pulse. So here you can see that you're making not only the hole that you made by photoionization, but you're making all these holes by electron impact ionization as well. And in addition, right, when you come with your probe pulse, what is it that you're actually seeing on your detector? You know that in the gas phase, you often have like quantum beats between two states that you've prepared in a superposition by your ultrafast pulse. But here, what are you going to see in the liquid phase? And so Robin and his colleagues, Swarnendu Bhattacharya and Ludger Inhester, calculated what the actual polarization response in the water sample would be. And what you can see here is quite interesting. So the actual response that you would actually see is, well, it's basically this this magenta curve for the polarization response. And what you can see is that it's confined to a very short time scale relative to what you might expect in a gas phase sample, where the polarization will last as long as that inner shell lifetime, which is on the order of 4 femtoseconds. So what this means is it's very important that we can actually, that our transient absorption snapshots with these attosecond time delays, they're actually meaningful on that same time scale, on that sub femtosecond time scale. And it's kind of interesting also, of course, that it's not always the case, but in this case, the condensed phase interpretation is actually somewhat simpler than the gas phase attosecond transient absorption. And this, this audience will all know from this very early work looking at real-time observation of valence. Hello. Okay, here. Let me hit that again before it stops. Okay, so, but you guys all know this, right? And I just wanted to say that it's kind of fun that it's actually, you think it's supposed to be really more complicated but it becomes a little bit less complicated because, okay, so here there was a strong field this was, this super famous experiment done in 2010 where there was strong field ionization of krypton and made this coherent superposition of these spin orbit states. And you could have absorption between that and excited states, and the difference between those absorptions was somewhere on the order of 0.7 eV, which you could then see that that energy splitting be reflected in this time oscillation that you could actually see, of course, both in theory and experiment. So that's kind of cool. But it's because, in the gas phase, you have these well-defined dipole oscillators and you have limited decoherence mechanisms. So you have all of these really nice oscillations that you can see. Okay, but for us, we don't have that. We have, distributed oscillators and they decohere very rapidly. And so what we see actually in the experiment, this is scanning from like 500 to 520 to 550-ish. And we scan the omega and the 2 omega simultaneously. And we can see in transient absorption, these peaks here and this big bleach here. This was done in 2022. And John, if you're online, you had the experiment like right after us, I think, right? And then we had the theory done by Lixing Liu and Xiaosong Li from the University of Washington. And because we know that, the snapshot is very rapid, we could actually just take a simple pentamer structure and calculate, oh, what do you see when you have the neutral and the ionized version? And, you can randomly distribute the holes in these two things, and you can calculate what you would see. And in fact, the theory basically reproduces what we see, including this bleach, which actually surprised us initially. We're going, what the heck is this big bleach, right? Turns out the big bleach at this pre-edge feature is due to a Stark shift induced by a neighboring valence hole. And so all of this was very nice and felt like it was, pretty tidy in some sense. Okay, so we were happy to see that this simple water pentamer can really model electronic structure changes that were observed. The pump probe times between these two are about 600 attoseconds. Okay, and so now what's interesting about this is, usually you have these ultra-fast studies and they're usually used to follow dynamics. But it turns out that in our particular experiment that we could actually do some static properties or have static properties revealed with this particular type of experiment, because we only had this one time delay at about 600 attoseconds. And it turns out that our particular experiment is actually the inverse of X-ray emission spectroscopy. And the X-ray emission spectroscopy has been used to infer the structure of liquid water. here is sort of a picture of the X-ray emission spectroscopy of water in the gas phase, liquid, and in solid form. And what you can see is that in the gas phase, there's sort of one peak. So, okay, so X-ray emission spectroscopy is when you've removed a 1s core electron, and all the valence electrons can fall back in and then produce a spectrum. And so it gives you some measure of that valence electron structure. You can see in the gas phase, you have the 1b1, the 3a1, and the 1b2. And in liquid phase, there's this funny splitting between this 1b1 peak, and then the 3a1 is kind of mushed in. And so it was hypothesized back in 2008 that this high resolution X-ray emission spectrum of liquid water was the signature of two structural motifs in ambient liquid water. And so the picture that was developed by Anders Nielsen and his colleagues was, oh, well, you know, what we have is kind of a low density liquid and a high density liquid. And the low density liquid are these, icebergs floating around in the high density liquid. And this is kind of what we have. So it's a little bit contrasting to the homogeneous tetrahedral structure of water that is mostly accepted. So, but also in 2008, there was another measurement of the X-ray emission spectrum. And it looked of liquid water, and it looked exactly like, the measurements were the same. It also had a split 1b1 peak. And instead, this group suggested, oh, maybe it's really just ultrafast dissociation in the core excited state that's actually causing this split in the X-ray emission spectrum. And so it turns out, right, that our experiment is basically the inverse of the X-ray emission spectrum. So in our experiment, right, what we do is we vacate these valence orbitals and then we promote back into them. And in X-ray emission, you vacated the whole, the core orbital, and then things fall back in. The difference, of course, is the X-ray emission takes place over on the order of 4 femtoseconds or so, whereas ours was this, ultra-fast sub-femtosecond time delay. And so, of course, in the X-ray emission spectrum, you see this doublet, but in our transient absorption experiment, we actually just see a singlet for this 1b1. And, this has been written about for a long time, starting out in, 2008, but it's been sort of a controversy around. And so as a result, there have been a lot of theories associated with this. And there are some most recent ones, one by Cruziero and Todd Martinez in 2003 on ChemArchive, and one by Takahashi and Harada in PRL in 2022 about, what is the X-ray emission spectrum? And as you can see, at 0 femtoseconds, they all have this sort of one peak, not so dissimilar from what we see. And at 4 femtoseconds, it develops this second doublet. And so it does appear that indeed the correct interpretation for the X-ray emission spectrum is that it's due to ultrafast dissociation in the core excited state and not due to two structural motifs. Thus, we have actually an experimental confirmation of this, resolution to this debate. Now, I just want to show you quickly, what this looks like. It's not your standard lab. This is what ChemRix at LCLS looked like in April 2022. The ChemRix is actually over here on the left. And the experimenters who were there taking the data in the middle of COVID, as you can see, right, are here. This is Stefan Muller, DJ Hoffman. This is my collaborator, Carolyn Pierce from PNNL, and this is my postdoc Schweili and student Kylie. And here they are at, and also Ming-Fu Lin, who's another beamline scientist, at the very end of the run at like 3 a.m. Carolyn said, oh, I should take a selfie to remember this, right? But without his mask, and the person who was doing lots of hard data reduction analysis was Schweili, and this is what he looks like. But of course, maybe it's not so nice just to have the faces of the experimenters, but also it's nice to show the faces of theory for this water work. And led by Robin Santra, there is Ludger Inhester and their postdoc Swarnendu Bhattacharya. And they're the ones who really showed how the response that you see in this attosecond transient absorption in the condensed phase is in fact confined to these very short timescales, which means that we can go, much further for many other time points, et cetera. And then for the theory that actually reproduced the results that we saw, it's Xiao Song's group with Li-Xing Liu doing much of the heavy lifting there. But of course, we can't actually do anything, right, unless we have these synchronized attosecond X-ray pulse pairs. And these were generated as is shown here. There's this so-called X-leap generation of the omega pulse in this first set of undulators. And then either with or without this delay chicane, you can make a 2 omega pulse in the second set of undulators, and you can actually measure what's going on by doing streaked photoionization with these two colors, and to have a circular VMI measurement of what's going on with the streaking laser. And this is how we actually knew what our relative jitter and durations and so on and so forth were. And the leaders of this enterprise were Ago Marinelli on the generation side, and James Cryan on the diagnostic side. And actually just this morning we heard that this work was accepted in Nature Photonics. So you'll be able to read all the details very soon about that. So let me just summarize here. Well, okay, so X-ray free electron lasers in general provide this new window for radiation chemistry. It's kind of our first opportunity to observe intra-spur dynamics. So a spur is when you have an initial ionization event and this little cascade. It happens all in, a few nanometer type volume. And we can also control the interspur dynamics because we can change the concentration of our little ionization events by changing the fluence of our X-ray pulse. Because we have this tunable X-ray source, we can start to identify radical formation mechanisms in solutions and in interfaces and really follow these reactions in situ. And I want to say that, it's really cool that these attosecond spectral snapshots via this attosecond transient absorption spectroscopy are really meaningful in condensed phase. And so our little situation was well modeled with a simple water pentamer, which I think is pretty nice. At least it's simple, something we can understand. And then finally, our all X-ray attosecond transient absorption experiment actually provided, some interesting information with respect to this misinterpretation of the X-ray emission spectrum and the structure of liquid water. So now we have many more collaborators than the faces I showed on that slide. This is slightly more complete. We have a lot of folks from Argonne. Also, we've enjoyed, collaborations with our CFEL colleagues for a long time. And at PNNL, they're super interested in looking at things that are kind of beyond water. And this is something that we have planned for the future. And we have many other collaborators as well. And of course, all of the folks at SLAC who really make this sort of experiment possible, not only for us, but for anyone who actually bothers to submit a proposal, get it accepted, and go and work with all these wonderful people. Okay, so then I think I'm done now, and thank you for your attention, and I'm happy to answer questions.
So thank you very much for this great talk. There is one from John, but he wanted to know what was the pump pulse intensity.
Oh, okay, so the pump
here.
So thanks so much for a great talk, Linda. I really enjoyed it, and it's great work. I'm asking this question about the pump intensity to understand how different it would have been to the pump intensity we used in our experiment a couple of
Uh-huh.
weeks later. Because what
Yeah.
we found is when you focus onto the jet, you
Yeah.
actually do create a significant amount of vapor phase water.
Uh-huh.
And we had to find a way to subtract that. We could do that in our measurements because we were doing intensity dependent scans. But I'm wondering whether or not you do not, I don't believe that you maybe can completely discount the presence of liquid of vapor phase
Well,
water.
okay, so we actually way defocused our X-ray pulses.
Oh, okay, that's great.
I mean, because we wanted to have, single photon and then so they were defocused, maybe to 100 microns or
Oh,
so.
okay. In that case, you were clean. That's good. Yeah.
Right. We were in a different regime. And then we selected the weakest pulses to do our experiment. Right. So we were pretty that was kind of in the design of the experiment because we wanted really isolated ionization events to look at.
Yeah. So what was the ionization fraction in the calculations? Because presumably there was some sort of assumption by Robin as he did the calculations for how much
Right. So eventually, so it was 1/10% for photoionization, but
Hmm.
then ended up with extra from the
Yeah,
electron
through the
impulse,
cascade, yeah.
right, from the cascade. So, but I'm really eager to hear about your experiments, John. I mean, I heard a
Well,
little.
I mean, hopefully I can share the results with you sometime. I mean, if
Yeah,
if
I
I
hope
get
so.
invited to one of these Atto Fridays to show it, I can do it even here. But
No, that would be great. I actually, uh, I think DJ and Jack, they showed me a little bit here and there, because they were wondering how what we were doing was impacting what they were doing, but they probably decided, oh, you're in such this different regime or
yeah,
something.
no, I mean, what I would say is two things. One is that we saw very clearly the Stokes signal we were looking for, and we just submitted the paper. Um,
Uh-huh.
but the other thing, which we maybe should talk about in the near future, is we've also effectively got an absorption measurement, but for a very short delay because it's inside a single pulse, so it's a 300 attoseconds, and I think they look similar to yours. So,
Oh,
so we should maybe compare notes on that and see whether one can't actually add an extra time, shorter time point to
uh-huh.
the data set.
Uh-huh. No, that
But
would that
anyway,
would be
we
really,
you and I should talk about that offline when
yeah,
we
we should talk
get a
about it
chance to
offline.
meet. Yeah.
Yeah, sounds great.
Yeah, are there further questions? If you would like to, there is one. Let me see. This is done. No, this was this one. So, I mean, people, do feel free to ask. Don't be shy. Uh, People are quiet apart from John. John, thank you so much for having you here. We should maybe schedule a talk. You were in our conference last summer, but you're always welcome to come and join. So, I mean, people are quiet, so let me ask one question. You mentioned that you were interested in going beyond water. What do you mean by that?
Okay. So it turns out that, well, maybe I can go to this. Okay, here's a slide. Right. Here's a slide about some of the motivations beyond water. So it turns out that there is this waste from World War II at Hanford that's primarily that's full of, bad stuff, but its main inorganic constituents are sodium salts of aluminates, hydroxides, nitrates, and nitrites. So all of these things, kind of low Z type things. And so now what you would like to understand, and what people do not understand, is when radiation hits these things, what are the reactive species that are formed? And so what we would like to do is to start looking at nothing this complicated, right? We would start with like salt. But how actually are these species formed? For example, when you form a quasi-free electron in water, it eventually, this is what we learned from our experimental theory thing in JAX about how electrons are actually solvated. So eventually they end up in a cavity. And in the cavity, the electron's in the middle, and the hydrogen atoms are kind of pointing toward it. But let's say you have like a sodium ion nearby, right? Does the electron go and reside near the sodium ion? Does that act like a new trap for the electron before it diffuses out into something much more complicated? And so it's kind of, we'd like to understand the real mechanism of, the electron, the fate of the quasi-free electron in complex environments, or more complex, but not this complex. But this is the overall motivation of things. And so you can really track what's going on with the electron, but also you can track what's going on with the hole. As I showed you, the hole is present in this water window. So anyway, this is one of the things. We actually have this very interesting experiment coming up at European XFEL, where we should be able to look at both the electron and the hole in liquid water produced by strong field ionization, but with a much better normalization than we had at LCLS. So we're kind of looking forward to that. They have a super fancy beam splitting off-axis spectrometer that we can use for great normalization. This is a super technical detail, but okay, it's something that would excite experimentalists, but not you, Carla. Right.
Yeah, I'm a theorist, but there are quite a few experimentalists in the audience, and we also have 10 people following on YouTube. I don't know if they're theorists or experimentalists, so I think they are probably excited, and it's good to hear anyway. One has to have an open mind.
Yeah. No, I mean, there are many things to do. I would say one thing is it's a little bit unfortunate that the configuration that we used for this experiment was not a standard one, right? This transient absorption where you basically disperse the radiation after it's passed through the sample. And it's not standard and probably isn't going to be available again for a little while until they have their more standard configurations sorted out. So, but eventually we will cross our fingers and we'll get it back.
Absolutely.
So,
yeah. So are there further questions? People are quiet today. Sometimes they are.
okay, well, it's a little
Anyone from my group who wants to ask something? No? No, they're quiet.
I'll ask.
Go, Tom, go.
You mentioned there was sort of controversy. Could you kind of speak to what are the sources of this controversy, maybe? Thanks.
And go back to this, right? So basically in super cooled water, there is a hypothesis that there are two types of liquids, a high density liquid and a low density liquid. So you know that ice, when you cool things, that ice is less dense than water, right? It floats, right? And the idea was that you have these two liquid phases that are high density and low density at low temperatures. And maybe we actually have those two phases also at ambient temperatures. And so this is what provoked this interpretation of this X-ray emission spectrum. And so they thought, oh, well, look, this 1B1 peak is like split into two. And so that means, we have these two structures. And one of them could be the low density phase that looks like ice. And one of them could be a high density phase that looks like water. And so they said, oh, okay, maybe that's what it is. And then other people came and did a bunch of other experiments saying, we think that it's actually due to ultrafast dissociation in the core excited state, right? So now this particular piece of evidence for the existence of two types of ambient water, this one particular piece of evidence is now no longer valid because it's both experimentally and theoretically disproven. So that's kind of what it is. I would say that that it doesn't mean that there are not two phases of liquid water at ambient temperatures. But it means this piece of evidence that they were using for it is not right.
Yeah, thank you. That was really interesting, actually.
Well, it is very interesting because so Anders Nielsen has been pursuing, it's quite interesting to go down to these super cold temperatures and high pressures where they think that they can actually see these two phases of liquid water. And so he's actually trying to do this with XFELs. And he has different ways so when you squirt the liquid into the vacuum, it cools, and it cools at a fairly rapid rate. It turns out that our particular experiment, it was still about room temperature-ish, right? But you can have the situation where it's much colder. And so he would try to cool it very, very cold and then also apply something to give it high pressure so that he could get to these they call it no man's land. This place, low temperature, high pressure, still liquid. It's a very strange place, right? But you can read about that in, I think it was also a science paper, maybe 2021 or something like this. But it's another XFEL type thing using these ultra-fast pulses. So,
yeah, thank you.
Mm-hmm.
Thank you. Any further questions? Okay. If not, we are going to stop, or if you want to use the opportunity to talk offline, we are going to stop the streaming. Because some people are shy, they always like to talk offline, but some people, they just hear. So,
Sure.
I mean, this is a great opportunity. Don't miss it if you would like to ask anything. So I'm stopping the recording, stopping the streaming. So for those on YouTube who are following us, take good care of yourselves. See you in 2 weeks.